Me of Science. Today we are going to discuss gene mutation. First of all, what is gene mutation? Mutation are changes in the genetic material such as DNA that result in the change in the protein. In gene mutation, the DNA code will have bases that are missed, added, or exchanged in a codon. Mutation occur at a frequency of about 1 in every 1 billion base pairs. Every body has about 6 mutations in each cell in their body. Now, mutations are not always seen. The affected gene may still function. It may be harmful, it may be beneficial, and it may have no effect on the organisms. How do mutations affect a population? Mutations are a major source of genetic variation in a population, resulting in increased biodiversity. For example, in a community we see people with different eye color, different facial appearance, and different face color. Some variation may help them to survive better. Now, how are mutations inherited? Mutations are only inherited from parent and they are transferred from parent to offspring, mean they are present in the germ cell. Mutation in the body cell only affect the organism in which they occur and are not passed on to the offsprings. Types of gene mutation. First of all, point mutation. The point mutation occur when the base sequence of a codon is exchanged. For example, GCA is changed to GAA. There are three types of mutation. First is substitution, second one is the deletion, and the third one is insertion. These deletion and insertion are combinedly called as frame shift mutation. Let's see an example of substitution mutation. Here we have a normal DNA sequence and this is the mutated DNA. As the name suggests, in substitution mutation, one of the bases in the DNA is substituted. For example, what has happened to the DNA? Here this adenine is replaced with this thymine. This is the substitution mutation. What will be the result? For example, if the CGA codes for alanine, TGC codes for threonine, and ATC codes for none. So if the mutation occur instead of A, T comes. So now the mutated DNA will codes for CGA codes for alanine, TGC codes for threonine, and this replaced DNA now will become TTC, it codes for lysine. In substitution mutation, a single nitrogenous base is substituted for another in a codon. It may or may not have the effect on the amino acid or protein. Now, what is insertion mutation? In insertion, a base sequence is added in the normal DNA. Say here, for example, here A, the nitrogenous base A is added in between T and G. What has happened to the DNA? And an adenine was inserted, thereby pushing all the base over a frame. This is an insertion mutation, also type of frame shift mutation. Here we see that CGA it is one codon, TGC it is a second codon, ATC it is an third codon. Now if a base come in between T and G, it will shift the whole frame. For example, now the CGA is one codon, the insertion, the red indicates the mutation, TAG it codes for another codon, and CAT codes for another, and this C will left alone. What will happen to the amino acid? The amino acid will change. For example, the TGC codes for threonine. When a base is added, now it will become TAG and it will codes iso leucine. A nitrogenous base is basically added or inserted into the DNA sequence in insertion mutation that causes the triplet frame to shift. It always affects the amino acid and consequently the protein. Now, what is deletion mutation? Here, in deletion mutation, one of the nitrogenous base is deleted. What has happened to the DNA? For example, this is CGA, this is TGC, this is ATC. Now, if one of the base, such as this base, is deleted, what will happen to the DNA? A guanine was deleted, thereby pushing all the base down a frame. This is called as the deletion mutation, also a type of frame shift mutation. Now look, this G is deleted and this C will come in place of G and this A will come in place of C to complete the codon. CGA codes for alanine. This TGC in normal DNA it codes for threonine, and this ATC will code for stop. While in mutated DNA, what will happen? CGA codes for alanine, this TCA will code for serine, and this TC will code none, and it will left alone. In deletion mutation, basically nitrogenous bases is added or removed from the sequences, resulting in the triplet frame to shift. It always affects the amino acid and consequently the protein. This is alanine, threonine and stop codon, while when one of the bases is deleted, so the new sequence will become alanine and this will code for serine. Which mutation would have the least effect on an organism? Surely it is a substitution mutation. The substitution mutation has the least effect because it changes only one amino acid, or it may change no amino acid. For example, in a normal DNA sequence, CGA it codes for alanine, TGC codes for threonine, and ATC codes for stop codon. If one of the base such as C that is cytosine is replaced with thymine, now this thymine will also code for a stop codon. In the second case, if this C is replaced with the G, now this ATG will code for tyrosine instead of stop codon. In case of stopping the protein synthesis. A common example of substitution is the sickle cell anemia that result in the change in the shape of the RBCs. Only one amino acid is changed in the hemoglobin that will result in different function, because in this case the shape of RBC is changed. The hemoglobin still function, but it folds differently, changing the shape of the RBC. Which mutation would have the most effect on an organism? Obviously it will be insertion and deletion. These have the most common effect on an organism because they affect many amino acids and consequently the whole protein. In case of this is the normal DNA, while this is the mutated DNA. Here we see that in the mutated DNA, when one of the bases is deleted, so the 3 codon is changed and it become now 2 codon. Similarly, in case of insertion, here in case of insertion we see that one of nitrogenous base will become extra. Gene mutation. Insertion mutation common example is Huntington disease. It will result in the involuntary movement and loss of the motor control that eventually result in the memory loss and dementia. The disease is terminal. Gene mutation. When does a gene mutation have the greatest effect on an organism? Obviously when it occurs in the germ cells such as gametes, it occurs in the gametes such as egg or sperm, or in early embryonic development. Now coming toward mutagens. What are mutagens? Mutagens are basically something that cause the DNA code to change. Change mean mutate. What are these? These include X-rays, chemicals, UV lights, radiations, etc. These are the basic work that are cited. Thank you for watching.